Hello everyone, welcome to today's live broadcast, Diabetic Cardiomyopathy Modeling and Screening with iPSC Cardiomyocytes. I am Judy O'Rourke of LabRoots and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Cellular Dynamics International. Cellular Dynamics is a leading developer and producer of fully functioning human cells in industrial quantities to precise specifications. CDI's proprietary products encompass multiple iPSC-derived tissue cell types that recapitulate native human biology. For more information on standard iCell and custom MyCell products, please visit www.cellulardynamics.com. We have a few important announcements before we begin. This webcast is designed to be interactive and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button lower left. I would now like to introduce today's speakers. Dr. Roberto Iacone is a senior principal scientist at Roche, Pharma and Research Development, Basel, in the Neuroscience, Ophthalmology, and Rare Disease DTA. He has a medical background and a strong research experience in stem cell biology. He established the stem cell group in the cardiovascular and metabolic discovery at Roche, where research in his group has been directed at understanding the pathophysiological mechanisms and development complications in the heart using patient-specific iPSCs, the patient in a dish paradigm. Recently, the group is establishing in vitro disease modeling to identify new drugs for the retina remodeling linked to age-related macular degeneration, AMD. His research interest includes the identification and characterization of genes that regulate tissue repair and regeneration, aiming to develop regenerative medicines activating endogenous tissue progenitors. Dr. Brad Swanson is Senior Director of Cell Biology Research and Development at Cellular Dynamics International, where he led the effort to develop the first commercially available HIPSC-derived cell product, iCell cardiomyocytes, and several other HIPSC models, including cortical neurons, dopaminergic neurons, skeletal muscle, cardiac progenitors, and astrocytes. Prior to CDI, Dr. Swanson was a senior scientist at Roche Nimmelgen, where he established, among other items, the industry's first sequence capture product for targeted next-generation sequencing workflows. He started his industry career at Promega as a senior scientist developing small molecule high-throughput screening products. Dr. Swanson received his PhD in cellular and molecular biology, cardiac differentiation from UW-Madison, undertook postdoctoral research in T-cell behavior at the National Jewish Medical Center, HHMI, in Denver, Colorado, and joined Columbus Children's Research Institute, Ohio State University Center for Vaccines and Immunity, as an assistant professor. I will now turn it over to Dr. Swanson for his presentation.
Okay. First, I'd like to thank everyone for making time to participate in our webinar today. I'm pleased to provide both a short introduction touching on CDI's technical capabilities in the iPSC space and some brief examples of disease modeling that were enabled by our products. After this introduction, we'll then hear Roberto's fascinating presentation on diabetic cardiomyopathy modeling with iPSC-derived cardiomyocytes. The agenda for the webinar is as follows. First, we will briefly cover CDI's product offerings and technological capabilities to enable iPSC disease modeling. Following that introduction, I will highlight disease modeling case studies in cardiac hypertrophy, Alzheimer's disease, and epilepsy where iPSC models from CDI were used. After that, Roberto will take over and describe his studies on diabetic cardiomyopathy using iPSC-derived cardiomyocytes. Deriving iPSCs and differentiated cells from iPSCs with a consistent high degree of quality is not a simple task, as many of you may know. This slide outlines the 4 critical areas of stem cell expertise CDI has spent years developing to enable delivery of human iPSC-derived products to our customers. These four critical areas are iPSC derivation, genome engineering, differentiation methodology development, and large-scale manufacture. Expertise in all of these areas are required in order for any company to provide robust iPSC-derived products to their customers. The discovery of iPSCs enabled a new era of personalized biology, which is being used to advance general and personalized medicine discoveries, both in the drug discovery and cell therapeutic spaces. To deliver products that enable rapid and robust advances in these areas, providers of iPSC-derived biological systems must meet the following requirements. Number one is a reproducible derivation of iPSCs from any donor, ideally in a parallel fashion. Number two, the ability to efficiently modify the genome of iPSCs to fix or induce mutations of interest or to create built-in assays. Number three, consistent manufacture of sufficient quantities of differentiated cells to support large-scale experiments, such as small molecule library screening. Number four, perform the first 3 points to tight specifications under a quality management system. We pride ourselves at CDI in delivering on all 4 of these points when developing and manufacturing our stem cell products. Over the past 6 years or so, CDI has commercialized nearly a dozen human iPSC-derived cell types, which are listed on this slide in chronological order. And these represent a large range of biological areas such as cardiovascular biology, with the iCell cardiomyocytes, and our recently launched iCell cardiomyocyte squared products, iCell cardiac progenitor cells, and iCell endothelial cells. Another area of significant focus is the neuroscience space with iCell neurons, iCell dopa neurons, and iCell astrocytes. We also offer our MyCell custom products, which allow the customer to have iPSCs reprogrammed, genome engineering performed, and differentiated cells made from their own donor or cohort of donors. The focus of today's webinar is disease modeling with iPSC-derived cells. Now, there are a number of strategies that can be employed to model disease with cells differentiated from iPSCs. Most of these strategies can be lumped into 3 categories: innate, induced, and engineered disease modeling. Innate modeling entails comparing the function or phenotype of cells from a normal donor with those from a donor with a genetically linked disease, such as Parkinson's disease in the case of dopaminergic neurons. Induced disease modeling involves culturing differentiated cells from a normal donor in healthy and disease-inducing conditions to study the difference in their function and phenotype. The recent development of several gene editing tools has opened a whole world of possibilities with engineered disease modeling. One can take normal donor iPSCs and create mutations of interest and then compare the differentiated cells from the normal donor and the engineered isogenic controls. The reverse can be performed as well, where one fixes a known mutation of interest in iPSCs from a diseased donor and then compares differentiated cells from the gene-corrected and original diseased donor iPSCs. Using this methodology in drug screening allows for what I like to call target-based phenotypic screening. One can have the benefits of studying a known target, but also have the added benefit of performing that study in the rich biological context of a differentiated human cell. Probing or quantifying that rich biological context can be performed with all of the typical analytical methods, such as flow cytometry, functional drug responses with cell-based assays, gene expression studies, high-content imaging, electrophysiological measurements, etc. The amount of published data demonstrating proof of concept of disease modeling in iPSCs is growing rapidly. This slide highlights just some of the wide range of disease areas where iPSC disease modeling has been successful. I'll focus on the cardiac and neuroscience areas for the remainder of the introduction. In the next part of the talk, I'll briefly describe 3 examples of CDI iCell products being used in disease modeling, starting first with cardiac hypertrophy. First, some background on CDI's iCell cardiomyocytes. They were the first truly commercialized human iPSC-derived product on the market, having been available since 2009. They're provided as a cryopreserved, greater than 95% pure population of cardiomyocytes. These cells are in use in 19 of the top 20 pharma and are a foundation for over 40 publications to date. Each vial comes with sufficient cells to plate an entire 96-well plate, as well as medium sufficient for culture for up to 2 weeks. The phenotype and function of iCell cardiomyocytes have been extensively studied in the key areas of cardiomyocyte biology, including metabolism and electrophysiology, and these cells are gaining significant adoption as a standard cell model to investigate cardiac toxicity. Studies comparing cardiac gene expression of multiple sources of primary cardiomyocytes versus isol cardiomyocytes demonstrate that isol cardiomyocytes have a gene expression profile much closer to native heart tissue than do isolated primary cardiomyocytes. Studies of energy metabolism and biochemistry demonstrate that cardiomyocytes exhibit robust mitochondrial respiration and have the signal transduction pathways one expects in a fully functional human cardiomyocyte. The ephys and contractile properties of these cells are also well understood and documented in the literature. The next few slides summarize work performed at CDI to demonstrate that isol cardiomyocytes robustly recapitulate a hypertrophic response in vitro. The top panel on the slide illustrates what happens to the heart and cardiomyocytes in particular when encountering a pathological hypertrophic stimulus, such as increased levels of endothelin-1. This response is characterized by increased cardiomyocyte size and protein synthesis and a reversion to a more fetal gene expression program. In particular, one can measure induction of brain natriuretic protein and atrial natriuretic protein in response to a hypertrophic stimulus. Straightforward culturing and treatment of isol cardiomyocytes with a hypertrophic inducer, such as endothelin-1, illustrated on the bottom of the slide, facilitates the study of this biological response. This next slide demonstrates that isol cardiomyocytes treated with endothelin-1 to induce a hypertrophic response upregulate BMP expression as a readout for hypertrophy. This mode of disease modeling would fall under what we call induced disease modeling, as you are treating what are normally healthy cells with a disease-inducing stimulus to induce the disease phenotype. This induction is robust and easily quantifiable using qRT-PCR for gene expression and high-content imaging, ELISA, and flow cytometry to look at protein expression. Each of these analytical methods has a wide dynamic range and highly reproducible technical replicates as demonstrated by the high Z prime values for the high-content imaging assay. The EC50s also have tight concordance across quantitation methodologies. There have been multiple publications using this model system to study mechanisms of cardiac hypertrophy. Now, CDI is participating in a large-scale collaborative project funded by the NHLBI to study the hypertrophic response of cardiomyocytes manufactured from iPSCs from a 250 donor panel selected from the Hypergen cohort, which is a population of African Americans known to have hypertension. GWAS studies have been performed on this group of individuals to study genetic risk factors of hypertension. CDI is enabling this study by deriving iPSCs and pure human cardiomyocytes from each of these 250 individuals, which will then be studied for their response to the hypertrophic stimuli in vitro. This study in disease modeling is a large-scale example of innate disease modeling, and that we are studying how the genetic makeup of the donors in the cohort influences the phenotype and function of their cardiomyocytes, differentiated from the iPSCs. CDI's unique capabilities and parallel manufacturing of both iPSCs and differentiated cells make large-scale disease modeling studies like these possible. For the remainder of my talk, the remainder of the introduction, I will focus on disease modeling in the neuroscience space, in particular modeling Alzheimer's disease and epilepsy. The last two examples of disease modeling in today's introduction both use our isol neuron product. These cells are terminally differentiated cortical neurons that exhibit a forebrain phenotype when examining gene expression of regional central nervous system markers. A particular note, as shown in the bar graph, these cells have a high level of FOXG1 expression. They exhibit a mature neuronal morphology and phenotype as visualized through immunostaining for beta-3 tubulin and MAT2 proteins. They're a mixture of inhibitory GABAergic and excitatory or glutamatergic neurons, with the population having a slightly higher proportion of inhibitory GABAergic neurons. Our neuroscience colleagues from GSK developed and published a nice model system to study Alzheimer's disease mechanisms in isol neurons using induced disease modeling. They demonstrated a dose-dependent toxic effect on isol neurons in response to amyloid beta peptide, which is known to play a role in the loss of neurons in Alzheimer's disease. This toxic effect was measurable both by loss in cell viability, measured by cell titer glow, and in reduction in average neurite length. Using this model system, they successfully identified a number of hits in a small molecule library screen that protected the isol neurons from the toxic effect of the amyloid beta peptide. Follow-up mechanistic studies helped identify the CDK2 as a target of one of the screen's hits, identifying a unique pathway in which amyloid beta protein exhibits its toxic effects to neurons. The last example of disease modeling is a case of engineered disease modeling that enabled the cellular study of epilepsy in vitro. And this study focuses on the sodium-activated potassium channel, KCNT1, which regulates neuronal excitability and determines how neurons respond to repeated high-frequency stimulation, which is really what happens during an epileptic episode. This ion channel has been shown to have a role in both cognitive defects and early onset childhood epilepsies. The genome engineering group here at CDI engineered a gain-of-function mutation in the KCNT1 channel at the P924L locus, or amino acid, and they engineered this in a normal donor iPSC line. And after they did the engineering steps, they differentiated cortical neurons similar to the isol neuron product from both the engineered and the unengineered lines, and tested these neurons for synchronous electrical activity in culture on a multi-electrode array instrument. The left half of this slide shows a 48-well plate from the Axiom Biosystems Maestro MEA instrument. Each of the wells on that plate contain a 4x4 grid of electrodes that measure single-unit recording, basically electrical activity, of the cells cultured on top of them. The raw data recording shown in the lower left of the slide demonstrates an example of synchronous bursting activity within one well, where a burst of electrical activity is seen at the same time across the majority of the electrodes in the well. Many quantitative parameters can be extracted from this type of raw data, and the right side of the slide touches on 3 parameters of importance for this model system. The bar graphs in white demonstrate that the number of bursts per minute in wild-type and KCNT1 gain-of-function mutant neurons are not statistically different. However, the intensity within those bursts are much higher in the mutant neurons when compared to the wild-type neurons, and this mimics the epileptic phenotype in vivo. This increased bursting intensity leads to a higher degree of synchronous activity in the gain-of-function mutant neuron cultures shown in the bottom bar graph. And then, to conclude, we demonstrated that this increased synchronicity can be attenuated with treatment by partial KCNT1 agonist quinidine in a dose-dependent fashion. And with that last data slide, I'd like to now hand the presentation over to Roberto for his presentation on modeling diabetic cardiomyopathy with iPSC-derived cardiomyocytes.
Thank you, Brad. Thank you, everybody, for attending the webinar on my side as well. And today, I would like to actually give you like an example how we use the iPS technology, and in particular the iPS-derived cardiomyocytes, for modeling diabetic cardiomyopathy. And in particular, I refer to the patient-in-a-dish approach that we use in PRED in Basel. And so before we start, I'd like to point that there are different ways to use stem cell in drug discovery. The first pillar on the left is as a tool. So now the patient iPS becomes an unlimited source of human somatic cells that are used for the patient-in-a-dish approach for the in vitro disease modeling. Then there is a second pillar that is the target, where now we try to develop small molecules to specifically activate our adult somatic stem cells that are in our tissue, and this is referred like chemical regenerative medicine. And then is the last one, that is the cell therapy, where now we inject cells to repair or regenerate the organ and tissue. In particular, in my group, and at Roche, we don't work on the cell therapy, and we have just few approaches in the chemical regenerative medicine. And today we will focus specifically on the use of the stem cell as a tool for disease modeling. And so I will try to introduce you a bit the problem, in particular highlighting the link between diabetes type 2 and the heart complication. Then we go to the model, the iPS-derived cardiomyocytes model that we use for drug discovery, and then hopefully we have some conclusion and some vision for the future. So let's start with diabetes type 2. This is clearly an unmet medical need as is becoming epidemic. In fact, by 2030, we will have almost like 500 million patients worldwide, and this number is increasing day by day. And the big problem is actually not that the patient they die for the hyperglycemia, but more for the stress that this diabetic microenvironment is triggering in different organ and tissue, and in particular in the cardiovascular system and on the heart. Just to frame how relevant is the myocardial complication of diabetes, you can, for example, you can imagine, and actually it's reality, that in the United States hospitals, for example, 1 out of the 3 patients that are admitted in emergency for heart failure, they will be diabetic. So there is a huge prevalence of in diabetic population of heart failure, and we know that this is actually almost like a primary condition that is called a diabetic cardiomyopathy, where there is a distinct primary pathophysiology in the cardiomyocytes of these patients. And on these slides, I'd like to point to you some important aspect of the pathophysiology of this disease. You can see on the left, for example, a post-mortem samples of cardiomyocytes from healthy patients. So the cardiomyocytes are very nice aligned, and there is no hypertrophy in the cells. Instead, if you take, for example, a post-mortem samples from a patient with diabetic cardiomyopathy, you can immediately see on the right that there is a disarray of the sarcomeric structure of the cardiomyocytes. They are completely scattered. And if you look on down, you see as well that the cell size is much bigger. There is really like an hypertrophy of the cells. So these important points are important to keep in mind, because I will try then to translate some of these phenotype in our in vitro approach. And so this is actually a bit what we try to do is to somehow resemble this diabetic stress in vitro on human cardiomyocytes, and then look if we can modify with drugs some of these molecular and cellular mechanisms that leads to the disarray of the cardiomyocytes and to the future of the diabetic cardiomyopathy. And of course, maybe it's important to say why we want to do that in a pharmaceutical drug discovery approach, and why it's important to do that. And now here in these slides, I refer to a nice review from our colleague at AstraZeneca, where actually they evaluated small molecules, the small molecules portfolio during the 2005 and 2010, and they somehow retrospectively tried to understand why there were like failure in the clinical phase. And you can see in the phase 2B, they had something like 88 compounds failing for efficacy. And now if you take these 88 compounds and you try to understand what is the reason for this failure, actually there is one of the main reasons was really the loss of and not a good link between the target and the models that they were using. So I learned a lot from this review, and so the key learning actually, in fact, that there is a strong link between, we need to have a very strong link between the target or the pathway and our disease, and we needed to somehow bring the human pathophysiology at the preclinical drug discovery in order to maybe achieve a better translation in the clinic. So with this spirit in mind, we tried to give now an example how we use the patient iPS in the diabetic cardiomyopathy. So first of all, there were already a lot of people working on this particular complication and this mechanism, and because it's very hard to have primary human cardiomyocytes, because of course you need to have a post-mortem access to the heart of the patient, a lot of people were using murine cardiomyocytes. We know that these cardiomyocytes respond differently to diabetic stressor, and they have a as well a different pathway and response. And the other big problem is that the murine models cannot account for the human genetic variation. So now the iPS gives the dual advantage that you can indeed having, for example, human genetic, having human iPS-derived cells, then you derive your cardiomyocytes, and then you try to mimic the stressor that we know very well in the clinics. And so with this in mind, we try to search for human cardiomyocytes that could be utilized in a pharmaceutical setup, so in big amounts. And at that point, we got in contact with CDI, and we actually evaluated their iPS-derived cardiomyocytes, and in particular, we could see that they had a very and good reproducible characteristic of the iPS-derived cardiomyocytes. They had all the features that were already highlighted by Brad. And the only things we needed to maybe to fine-tune for our approach was to induce maybe a more mature phenotype. Indeed, it's possible to manipulate the metabolism of the iPS-derived cardiomyocytes to induce a more mature phenotype. And we actually stealed some trick from nature. In fact, during our fetal life, the cardiomyocytes will use mostly glycolysis for energy production, and then as soon we are born, they will switch instead to fatty acid beta oxidation for energy source. So with this in mind, we try to do exactly the same. We plate the cells in a normal medium, we let them attach for 2 days, and then at day 0, we culture them with a maturation medium that is completely glucose-free, is completely chemically defined, and this has energy source of fatty acid. So we force the cells to become and to go in beta oxidation and eventually to mature. Of course, we needed to convince ourselves that indeed we have a more mature phenotype, and we needed to convince as well our reviewers in the paper that was published recently in Cell Reports. And we used it for these 2 parameters that are very well described in this review from Charles Murray. The first parameter is actually the sarcomere length and the sarcomere organization. So for example, if you look at the sarcomeres in the alpha-actin, with an alpha-actin string, you will see very nice this striation, these stripes in the cardiomyocytes. And the other one is instead a parameter in the electrophysiology. We know that, for example, cardiomyocytes, when it's more mature, will have an increase in the sodium currency, and this will increase the duration of the action potential and the upstroke velocity. So we took all these parameters and we started to ask, do we indeed, after this maturation step, induce this, reach these parameters? And for the sarcomeric organization, we could indeed confirm that after 3 days in maturation medium and forcing the cardiomyocytes in beta oxidation, we could have an increase of the cardiomyocyte score that actually reads the striation in the alpha-actinin channel, as you can see in this picture, and as well we could show that there is an increase in the sarcomere length, as shown by the reviewer and by other papers before. And this translates as well to an increase of key genes that are more expressed after the 3 days of maturation medium. So the next part was indeed to confirm the electrophysiology and the sodium currency. So for the sodium current density, we use a voltage dye for the sodium. We could see indeed that there is an increase of the upstroke velocity, of the upstroke velocity here, and as well of the duration of the action potential. This was as well very well confirmed by the expression of gene expression of some key sodium channel, and this is the final proof by patch clamp. We could see indeed that there is in red after the 3 days an increase in the sodium currency and even if it's not statistically significant, but we could see that there is higher conductance for the sodium currency. So with this, we were very well convinced that at least we could have a more mature phenotype of the cardiomyocytes. And then we went to the second step, that is actually the mimicking of the diabetic stressor in vitro. So for this regards, now after the 3 days in maturation medium, then we switched to a medium that has some of the key diabetic stressors that are reported in the literature. In particular, high glucose. We know very well diabetes is very well described by hyperglycemia, of course, increase of insulin, increase of fatty acid, and some neurons, some hormones like the cortisol and the endothelin one, that indeed are higher in the plasma of patients with diabetic cardiomyopathy. So when we expose the cells to the diabetic stressor, we could observe different phenotypes. So the first one is secretion of the BMP biomarker in the cellular medium to very high quantity, so 200 picogram per ml. And I like to point here that indeed, for example, when you recruit in clinical trials for heart failure, one of the inclusion criteria is having plasma BMP higher than 150, 180. And very importantly, we could see that after these 48 hours in diabetic stressor, we could see a reorganization of the sarcomeric structure, an increase of hypertrophy at the cellular level and at the nuclear layer, and very important that the structural disorganization was connected to a decrease in the calcium transient, meaning that there is a correlation between the structural cellular organization and the calcium transient. Not only we could see this, but we could see as well a different phenotype of the diabetic structural remodeling. For example, lipid accumulation with the nail-red assay and the oxidative stress lipid peroxidation with a specific dye. Once we did this, to be really sure that what we see in the dish is somehow resembling what is happening in the cardiomyocytes of this patient affected by the diabetic complication, we gave some of the samples to one of our collaborators, Professor Henning Stenberg at the University of Basel, and we performed electron microscopy. We could see that, for example, cardiomyocytes in maturation medium has a nice maturation of the sarcomeric structure. In particular, you can see the Z line here. And now, if you indeed culture the cardiomyocytes under the diabetic stress, you see a completely disarray, a scattered morphology of the sarcomeric structure, and you can start to see these Z bodies. So these Z bodies are very important for the characterization of the model because they've been already described in mouse models of heart failure, like the T modeling knockdown, where you can see this formation of the Z body, and more importantly, in post-mortem samples with the patient affected by diabetic cardiomyopathy. So with this, we were really convinced that our in vitro surrogate was resembling some aspect of the clinical feature of this patient, and we could really identify three major parameters that we wanted to follow in our later on drug discovery approach: the BMP release in the medium, the hypertrophy as the cell and nuclear layer, and the sarcomeric disorganization. So now the next step is to translate these endpoints in a phenotypic drug discovery approach. Before doing that, we tried to understand a bit what was going on at the level of the pathway and the genes. And to do this, we performed a genomic analysis where we compared the cardiomyocytes under maturation medium and the cardiomyocytes under the diabetic stressor. We could see, for example, that upon the diabetic stressor, we can activate in red pathways that are indeed correlated with the glucose metabolism and the Krebs cycle, so showing that indeed the cells were going back in a sort of glycolysis. And very importantly, we could see downregulation of pathway, for example, in the protein translation, in unfolded protein response. So very interesting. Now, what you can do, and you can see these data that are published, you can go in one, for example, of these dots and retrieve all the genes that are up and down regulated in this particular pathway. Now, having this list of genes, you can see if there are any publicly available library or Roche library compounds that are having small molecules that are annotated versus these particular genes. So we did this in particular with the publicly available library, and we could then assemble a library that could be tested in our phenotypic screening approach with our iPS-derived cardiomyocytes. And the screen is actually performed that the cells are kept for 3 days in maturation medium, and then at day 3, we now apply the compound and the diabetic stressor at the same time, and we read after 48 hours incubation. Very importantly that for each well, we record the 3 endpoints. So the cardiomyocyte score, so the organization of the sarcomers, the BNP release in the medium, and the nuclear layer of the hypertrophy. And you can see that in all 3 parameters, we could see a decrease or an increase of the signal. And based on the fact that we have seen in this genomic analysis that we have seen upregulation of the endothelin pathway and of the cortisol pathway, we decided to use as positive control a combination of mifepristone and bosentan. Bosentan is an endothelin-1 inhibitor, and mifepristone is a glucocorticoid inhibitor. And indeed, in all our plates, in all our screening, this combination, this positive control, could reestablish all our endpoints, as see here in the orange bar. Based on this, we then performed the full pilot screen with 480 compounds, where we have, as I said, a very nice annotation of the target. So you can imagine that this is a sort of a chemical biology approach. And out of 480, we could indeed identify the 47 hits. Then, as you will do in any screening campaign, so we confirmed these hits with a dose response. And you can see that we have different compounds eating different pathway. For example, a lot of compounds were modulating the calcium signaling, other were kinase inhibitor, other were modulating the protein translation and protein processing control. And of course, these were selected based on our genomic analysis. And we had very good results, in particular in compound modulating the calcium signaling and some kinase inhibitor, multikinase inhibitor, and as well compound modulating the protein translation and protein processing. So now what is very fascinating that different compound with different mode of action can indeed, in a dose response, revert these endpoints that we addressed with our screening campaign. The next part was really to see if any of these hits could indeed as well induce some or revert some functional endpoints. And for example, before we look at the lipid accumulation and peroxidation, you can see that some calcium modulating like the fluspirilen can indeed decrease the lipid accumulation and can as well decrease the peroxidation, even if the mode of action is completely unrelated to peroxidation and lipid accumulation. And very interesting, for example, compounds that are involved in, for example, protein translation can restore, for example, the calcium transient that they are disrupted by the diabetic stressor. So now this is actually opening a completely new chapter for us, because we want to understand at the molecular level what these compounds are doing. So how do they change the molecular and the pathway context of the cells, and in particular why compounds that are eating different targets can induce the same phenotype, like for example, compound that they modulate the calcium signaling, and compound that, for example, modulate the protein translation. But this is working ongoing, and we will hopefully report something during this year or next year. The next part of the talk is really now focusing on exploring at best, the iPS technology, and in particular to trying to merge in vitro the environmental driven stress that we did with the diabetes stressor and the genetic background of the patient. So diabetes type 2 has a very heterogeneous genetics, and even more heterogeneous is the genetics of diabetic cardiomyopathy. So we could not identify a single genes with high penetrance. And for this, we tried to have a more an empiric approach, where we selected the patient with the extreme phenotype. So what you will see in the clinics, for example, a fast progressor, a patient having diabetes type 2 and progressing extremely fast to diabetic cardiomyopathy or cardiovascular disease, or on the other hand, another patient that is called a slow progressor, so a patient that is having diabetes type 2 since many years, but is not having any clinical symptoms of the cardiovascular complication. So, and with this approach, we, together with our colleague at MGH in Boston, we selected out of different patients 2 extreme patients. One in blue, that is the diabetic type 2 slow progressor, that, for example, despite the fact that there's having diabetes type 2 more than 15 years, having a family history of diabetes, had no sign of CVD. And on the other hand, we have the red patient that is the opposite. So it's actually a patient having diabetes type 2 less than 10 years, but having a strong penetrance and a clinical documented cardiovascular disease phenotype. Of course, the advantage of this model is that we can later on compare the medical history to our in vitro phenotype, but the key disadvantage is that we are dealing with an extremely small number of patients, so this needs, of course, further confirmation. So now the great advantage that once we obtain the iPS-derived cells from this patient, we could send the iPS to our colleagues in Wisconsin at CDI, and with the exact same protocol, they differentiate the cardiomyocytes as we used before in our standard cell line to establish our protocols. And once we obtained at Roche the iPS-derived cardiomyocytes from this patient, we had a big surprise. Actually, even without any diabetic stress, so at the baseline, just in the maturation milieu, we could see that the diabetic patient, the slow progressor and fast progressor, had a less organized structure in the sarcomeric structure. And we could see as well a difference between the slow progressor and the fast progressor. In fact, at baseline, without any diabetic stress, or the fast progressor, for example, in red, had already a reduction in the calcium transient frequency, and some work that was done even at Wisconsin at CDI, you could see that this patient had more irregularity in the beat rate using the impedance readout with the exergens. So this was extremely, fascinating. Of course, now we don't know what are the reason of this difference, and we believe that there is something at the genetic level, but we don't know yet what it is. Not only we could see this phenotype, but we have seen as well a difference between slow progression and fast progression in terms of lipid accumulation and oxidative stress. The last part, is of course to see if some of the compound that we identified could reverse some of this phenotype. I like to stress that again, in this, we tested before the compounds without any diabetic stressor. And this is very important to keep in mind because you can see that, for example, already the positive control, so the bosentan and mifepristone, could indeed reverse, for example, the cardiomyocyte score that was lower in the slow progressor and in the fast progressor. For the slow progressor, in blue, we could even revert completely the phenotype equal to our standard line, and we could revert as well for the fast progressor, but with less success. So this is a very important point because, first of all, here we don't have any diabetic stressor, so they are not under cortisol and endothelin 1. But of course, the two positive control, the bosentan and mifepristone, they indeed already work on these cardiomyocytes. And later on, we have shown that this particular patient, in particular the slow progressor and the fast progressor, have already a baseline, so without diabetic stress, already an upregulation of the endothelin pathway at the molecular transcriptomic level, and as well of the glucocorticoid pathway. And the other big difference, again, that we can see that despite these cardiomyocytes, they have the same maturation, we have seen, again, a different profile in response between the slow progressor and the fast progressor. And indeed, if now we take, for example, our 18 hits that represent a different mode of action, we could again confirm the difference in response between the slow progressor and the fast progressor. In the slow progressor, actually 14 out of 18 can revert the cardiomyocyte score, and in the fast progressor, just 10 out of 18 can revert this, showing that the fast progressor has again a more prominent problem in at the molecular, at the cellular level. It's interesting to see that, for example, the top compound like the psigargin and fluspirulane are positive for the slow progressor and the fast progressor, and they regulate, the calcium signaling in the machinery, or they are related with the calcium signaling machinery. And this is very nice as well because actually in different in vitro disease modeling approach that are published by very good labs, in particular on the delayed cardiomyopathy and hypertrophic cardiomyopathy, where now the investigator induced the in vitro phenotype mutating some specific genes that are associated with this inherited cardiomyopathy, and not by chance, but they have seen similar phenotype that we have seen under the diabetic stress, in particular the hypertrophy, the sarcomeric disorganization, and as well dysregulation of the calcium signaling. And not by chance, but for example, some of these phenotype could be reverted by the use of channel calcium blocker like verapamil, nifedipine, and we have found indeed that, for example, flulispilene is as well a modifier of the calcium blocker. So what I want to say that there is a nice analogy between our approach that is in case of exposure of cardiomyocytes under drug under the diabetic stress that is environmental driven, and this kind of publication where instead we have a genetically driven phenotype. Now with this I like to conclude and give space maybe for a bit of discussion. So I hope that I was able to show you that the way we think about using the iPS technology for drug discovery, and in particular that we believe that placing the human pathophysiology at the core of our preclinical drug discovery will have huge impact in the future. In particular, we can maybe early on eliminate toxic compounds that can have some safety concern later on in the clinics. And we believe as well that this approach can have a huge impact on betting on drugs and eventually new drugs that will have a better efficacy in the clinical trial. So circumventing some of the problems that I highlighted or tried to highlight with the review from our AstraZeneca colleague. Of course, this process needs to be proven. It's not yet, so we don't have yet a drug in the clinics that was discovered with this approach, but it's definitely a space that needs to be watched very carefully, because as Brad as well showed you, it's used in different fields and by different people in science. So I have my big hope that will bring a lot of results. And with this, I need to acknowledge all the people that worked on this particular project, in particular Faye and Stefano, that were two postdocs in my group. They did incredible work. And then as well from my group, Fred and Jack, that started a lot of this approach in the lab. Benjamin for the electrophysiology and validation of the cells. Chiara for her discussion and a lot of advice on how to perform this approach, and the team of Laura and colleague here at Roche that analyzed a lot of this genomic data. Very important to say that we had a fantastic collaboration with people at CDI, particularly Kyle, Blake, Kobe, and Asuyo, so we worked very close to them. Even sometimes they performed experiments with us, so it was great. I already mentioned Henning and Alexandra for the electron microscopy, and then I like to conclude acknowledging the people in Boston in the MGH that were generating the IPS lines. And with this, I conclude, and I'm very happy to take your question.
Thank you for the informative presentations. Before we get started on the question and answer session, I'd like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. And our first question is, let's see, this question is for Dr. Swanson. Can I-cell cardiomyocytes be used for patch clamp studies, specifically sodium ion channel analyses?
Yes, so as Roberto pointed out in some of his slides, you can use isocardiomyocytes for most analyses of the main ion channels active in human cardiomyocytes, sodium, potassium, calcium, and the various variants of those channels. So these methodologies have been well worked out and well published and very well studied, not only in normal cardiomyocytes, but in disease patients as well.
Another question for Dr. Swanson. Do you perform custom mutagenesis on genes of interest in the iPSC-CM?
So yes, we do. We perform the mutagenesis actually in the iPSCs as part of our MyCell services. And we use either CRISPRs or TALENs, or nucleases, to facilitate genome editing or genome modification. And then we'll go ahead and differentiate those cells with the modified genomes into the cell type of interest. So that is one of the services that we offer in our MyCell product line.
This question is for Dr. Iacone. Can you comment on the drugs and their pathways that work with SP but not FP?
I'm sorry, could you repeat the question? Sorry, I could not hear.
Yes, of course. Can you comment on the drugs and their pathways that worked with SP but not FP?
Yes, so yeah, it's a very good question, and at the moment we don't have the final answer. What I can say that modulating the calcium signaling seems to be a very powerful approach, both with the slow progressor and the fast progressor. It seems that the fast progressor is a very somehow very hard patient to treat, and now what is very interesting is to see if actually in both slow progressor and fast progressor, if the addition of the compound is indeed inducing the same molecular signature. And this is what we are doing with other colleagues here at Roche. But at the moment, we don't have yet the answers. Another approach that is very interesting and we try to do is, for example, overexpress the SERCA2, because this is an approach that is being evaluated by different companies for gene therapy, and it will be very interesting is to see if actually such overexpression can rescue our fast progressor phenotype, but we don't have yet this answer.
Another question for Dr. Iacone. Was the cell line to cell line heterogeneity excluded in analyses of diabetic iPSC derived cardiomyocytes? And does the presence of disease phenotype in diabetic iPSC derived cardiomyocytes suggest the genetic origin of the disease?
Yeah, so I don't know if I understood the question correctly, but I think about the heterogeneity, I think you refer to the fact if we have just cardiomyocytes in our culture, and indeed, yes. So the prevalence of the culture, more than 95% is cardiomyocytes. So we believe that the effect that we see in the dish is primary and cell autonomous due to the cardiomyocytes. About the cause of the reason for this difference between, for example, the fast progressor and the slow progressor, and the fact that we see with the patient IPS already a baseline a phenotype, we believe that is genetically, because we know very well that after reprogramming we somehow remove epigenetic or remove partially the epigenetic fingerprinting. So we believe that it's more genetically, but as a lot of people already show, it's very hard to fish the genetic cause of this predisposition, because most likely there were different genes involved. And to be honest, we are more focusing on understanding, the response of this patient IPS to the drugs in order to somehow evaluate some new therapeutic targets.
Okay, another question for Dr. Iacone. Regarding diabetic CM phenotype, it seems that the patient-specific phenotype is intrinsic to CM, not dependent on diabetic milieu, while in normal iPSC CM, it is the diabetic milieu that induces phenotype. In other words, Does this mean that the diabetic milieu in vivo does not contribute to CV phenotype?
Yeah, I think it's so. First of all, I think the two things are connected because, as maybe I just quickly refer to this, but when we analyze when we perform genomic analysis of these patient iPS cardiomyocytes, we could see that indeed they had dysregulation in the endothelin pathway and in the glucocorticoid pathway. So, and we didn't know this before, you know. So there is a connection, and I think, you know, like every disease is not like, we cannot say it's completely genetically or it's completely diabetic, it's completely environmental driven, but it's always a combination of the two. So most likely these patients, they have a predisposition, you know, to genetic predisposition to overreact to the diabetic stressor, and these together induce the phenotype. So I think it's very, we cannot, you know, really frame the genetics or the environment, but it's always a combination of the two. And I think our approach, even if with a small number of patients, is really to highlight that with the patient iPS, you can do this kind of approach. You can have a genetic predisposition with you, that you bring with the patient iPS, and on top, you somehow investigate the response to the clinical environment that can be, for example, in this case, a diabetic stress.
This question is for Dr. Iacone. How would one move from a hit in a phenotypic screening to identifying a target? Is this necessary for further drug development?
Yeah, so this is, maybe it's a guy from Pharma, because this is a very good question. So honestly, there is not, so no one, so the FDA will not require you to show the target for your compound. They will just ask you to show that the new drug works. But in reality, you know, for the development of these new potential drugs, it's very important to know the target, because you can work better. For example, if you see some safety related issue, or if you want to understand better which is the therapeutic index and so far. Now, how to find this target after a phenotypic approach is very challenging, and we try different approach for different projects. And in reality, it's very hard to have the single gene. You have, utilize a different methodology, for example, transcriptomic, proteomic, or binding of the small molecules to a protein. You will have always a different list of genes. And this is very, it's very informative, but it's clear that you usually don't get a single target. So this is a bit the challenge that you get, when you go in phenotypic drug discovery. But this is as well the advantage, because, you want to go for somehow a more complex effect. So honestly, I don't have the final answer to this question, but I think that it's important to have like a balance in your portfolio between phenotypic drug discovery approach and target-based approach, because it's clearly shown that some great drugs comes from a purely target-based approach, and some they come from the phenotypic drug approach. And if you have like at least an idea of the pathway that you modulate, is very important for clinical development.
This question is for Dr. Swanson. Have either of your organizations done any disease modeling in 3D?
I'm aware of a number of customers or collaborators that have cultured our cardiomyocytes and our isocardiomyocytes as well as isohepatocytes for separate reasons in 3D culture systems to study how that changes the biological response to compounds, et cetera. So yes, I'm aware. We're not doing it actively internally very much, but we know a number of our customers as well as some other collaborators are studying the biology of a number of our cell types in 3D culture.
I would like to thank our sponsor, Cellular Dynamics International, for making today's educational webcast possible. Today's webcast will be available for on-demand viewing through October 2016. You'll receive an email from LabRoots alerting you when this webcast will be available for replay, and we invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time. Goodbye.